हेलो एवरीवन एंड वेलकम बैक टू माय प्लेलिस्ट ऑफ माइक्रोबायोलॉजी जो हम लेविंसन लेंज पब्लिकेशन से कर रहे हैं। चैप्टर नंबर 11 आज डिस्कस करेंगे और ये चैप्टर है अबाउट एंटीबैक्टीरियल रेजिस्टेंस। वेरी इंपॉर्टेंट टॉपिक बिकॉज आई मीन अ नाइटमेयर वुड बी अ सिनेरियो जिसमें यू हैव द आर्मामेंटेरियम, आपके पास एंटीबायोटिक्स हैं और ये जो बग है जिसके अगेंस्ट एंटीबायोटिक्स हैं वो एंटीबायोटिक्स अब इफेक्टिव नहीं है। सो दैट्स गोइंग टू बी अ नाइटमेयर फॉर एनी क्लिनिशियन। आप ज़रा सोचें ना कि हमारे पास जो माइक्रो ऑर्गेनिजम्स हैं, पर्टिकुलरली इस चैप्टर में हम चूंकि बैक्टीरिया की बात कर रहे हैं, सो थिंक अबाउट एनी बैक्टीरिया, ट्यूबरकुलोस बैक्टीरिया फॉर एग्जांपल, माइक्रोबैक्टीरियम ट्यूबरकुलोसिस which causes TB. Now we have a set number of drug which work against tuberculosis. Now that would be again, as I said, a nightmare for a physician or a clinician कि जो drugs हमारे पास मौजूद हैं, drug A, B, C, whatever, all of them are not working against TB. Imagine that day how infectious that person who has tuberculosis infection would be. Because now you have no way to treat tuberculosis and the infection will be spreading and spreading and spreading. So antibacterial resistance or antimicrobial resistance is such an important topic because and this is happening. I mean, there are so many organisms now against which drug resistance is such a common problem. Even for tuberculosis there is like multi-drug resistant cases of TB. There are total drug resistance cases of TB also been reported. So that is, a very, very worrying condition. काफी परेशानी की बात है ये. So इस chapter में what we are going to discuss is some basic principles of antibiotic resistance. So there are basically 4 major mechanisms. When we talk about resistance, so एक आपको idea होना चाहिए कि the drug that we use, so invention of the drug, formulation of the drug, that's where you are playing smart, where humans are super smart, they're making drugs. And when there is resistance development, it's the microorganism which is now the smart guy, because this microorganism is generating some mechanisms जिससे वो इन ड्रग को डील कर रहा है, इन ड्रग से बच रहा है, इन ड्रग से रेजिस्टेंट हो रहा है. सो वी आर टॉकिंग अबाउट माइक्रोबियल स्मार्टनेस इन दिस चैप्टर. सो देयर आर 4 मेजर मैकेनिज्म्स दैट मीडिएट बैक्टीरियल रेजिस्टेंस टू द ड्रग्स और वो 4 मैकेनिज्म्स हैं. नंबर 1, बैक्टीरिया में प्रोड्यूस सर्टेन एंजाइम्स दैट इनएक्टिवेट द ड्रग. So it's a simple method, you see. Simply bacteria they have learned. So if this is a bacteria and there is a drug A used to work against this bacteria, but now when the drug enters, bacteria is producing a particular enzyme which will make drug A ineffective. So that's the first mechanism, that bacteria produce certain enzymes which are actually inactivating the drug. Examples being, so that's the first category, inactivate the drug. Or important example is cleavage of the drug by beta-lactamase, which is produced by certain bacteria. Okay, and the drugs which are commonly affected by this enzyme are the penicillins, cephalosporins, and carbapenems, which are the beta-lactam drugs. So they contain beta-lactam ring, and the bacteria is producing beta-lactamases. It destroys the drug. The second mechanism is that bacteria synthesize modified targets against which the drug has a reduced effect. So in this category, what happens is, suppose this is a bacterial cell, and that's a drug A. The drug A enters, and the drug A, say for example, has to work on this cellular target. Okay, so that's a bacterial protein or anything जिस पर जाकर drug A ने अपना action करना है. Now the bacteria becomes smart, and what the bacteria does now, instead of producing this rectangular target, it now produces this star-shaped target, and drug A is not able to bind to this new target effectively as it used to bind to the rectangular target. So by changing the structure of the target, effectivity of the drug is changed. So that's another method, and this is modify the drug target in the bacteria, and there are so many important examples for this, such as penicillin binding proteins. So penicillin can no longer bind to its target proteins. Mutations in proteins in 30S ribosomal subunit, aminoglycosides, and streptomycin can now no more act on those subunits. Replace alanine with lactate and peptidoglycan in the cell membrane, and vancomycin cannot work on this alanine added peptidoglycan. If there is mutation in DNA gyrase, quinolones won't work because they work on gyrase, and the gyrase changes after mutation. So the quinolones entering into the cell worry कि भाई गैरेज है किधर, मैं कहाँ जाके अपना एक्शन करूँ, बिकॉज उसमें तो म्यूटेशन हो गई, उसका टारगेट ही चेंज हो गया. म्यूटेशन इन RNA polymerase, rifampin, mutations in catalase peroxidase system, isoniazid won't work. So this is such an important table, you see. The third mechanism by which bacteria generate resistance include reduced permeability to the drug, such that an effective intracellular concentration is not achieved. Now, what's happening in the third case? So that was the first case when the drug was being inactivated. This was the second case when the target in the bacteria was being changed. Now, the third scenario. The third scenario is this is a bacteria and that's the drug. Now, drugs usually enter the bacterial cell by using certain forms of receptors, and they attach to the receptor, get internalized, that sort of thing. But bacteria, with the passage of time, may also learn to change the permeability patterns. So now the drug is not able to enter into the bacterial cell effectively. That's the third category. And reduced permeability of the drug, such as if there is mutation in the porin protein. Now imagine porin proteins are these proteins which allow the drug to enter. If there is mutation, the structure of the porin protein is changed and now the drug can no more enter into the bacterial cell. और जब enter ही नहीं होगी तो घंटा उसने काम करना है, that sort of thing, okay? And this type of mechanism affects penicillins, aminoglycosides and so many other drugs as well. Now, the fourth mechanism again is very interesting because now the bacteria learns to throw the drug out of the cell and they develop special pumps which are regarded as multi-drug resistant pumps. So what basically happens here in the fourth scenario is that here is the bacteria, the drug enters, and now the bacteria catches the drug, throws it out again. Get out of my body. And these are the drugs which are affected by this fourth mechanism. So अभी जो पूरा बाकायदा चैप्टर हम पढ़ेंगे, पूरे का पूरा चैप्टर 11, उसमें इन्हीं 4 mechanisms के अराउंड सारा डिस्कशन होगा. So keep these 4 mechanisms in your head and the whole discussion will actually be revolving around these 4 concepts, okay? This is a must-do table for bacterial resistance. Most drugs resistance is due to genetic change in the organism, either a chromosomal mutation or acquisition of any plasmid or transposon. So basically the genes are changed. So if you imagine there is an extra enzyme production which destroys, चाहे आप mechanism 1 की बात कर रहे हैं, so there is an addition protein which is going to be produced, और इसके लिए भी आपको genetic change चाहिए. Modification of the target, which was the second sort of mechanism, उसमें भी आपको genetic change चाहिए. देखें, sequence of DNA change होगा तभी different RNA बनेगा, RNA different बनेगा तभी different target protein बनेगी. So there is a genetic change involved in almost all of them. The term high level resistance and low level resistance ये अक्सर यूज होती है clinics में. So let's have the concept. What does that mean? The term high level resistance refers to resistance that cannot be overcome by increasing the dose of the antibiotic. So this is really serious type of resistance. आपको different antibiotic use करनी पड़ेगी. You will have to use a different class of antibiotic. And low level resistance is basically, it refers to resistance that can be overcome by increasing the dose of the antibiotic, okay? Hospital acquired infections are significantly more likely to be caused by antibiotic resistant organisms than the community acquired infection, because hospitals में आप ऐसे समझ लो ना कि बड़े ढीठ किस्म के बैक्टीरिया होते हैं जो ऑलरेडी काफ़ी रेजिस्टेंस पैटर्न डेवलप कर चुके होते हैं, बिकॉज दे आर लिविंग इन अ हॉस्पिटल ना. सो हॉस्पिटल में एंटीबायोटिक्स हैं चारों तरफ, पेशेंट्स हैं, सो दे आर nasty bugs, you know, they already have developed a lot of resistance. And this is especially true for hospital acquired infection caused by Staph aureus, enteric gram-negative rods. So basically a generic principle that hospital acquired infections are more prone to resistance. 2 types of gram-negative rods are especially important in hospital acquired setting of multi-drug resistant gram-negative rods and extended spectrum of beta-lactamases, ESBL जिनको हम कहते हैं, producing gram-negative rods. MDR negative rods produce beta-lactamases that inactivate the bacteria, and ESBL gram-negative rods produce beta-lactamases that inactivate second, third, and fourth generation cephalosporins. So pretty hard bugs to treat. Okay. Now, some important bacteria and their resistance. This is an important table as well. You have to consider gram-positives and gram-negatives, and in both the categories, cocci and the rods, and then also the mycobacteria. So Mycobacterium tuberculosis, for example, and the clinically significant drug resistance has been reported in isoniazid and rifampin. So these bacteria are, बल्कि मैंने इसमें आपको बताया कि now we also have multi-drug resistant tuberculosis as well as total drug resistant tuberculosis. And if you talk about the other species, Mycobacterium avium intracellulare, these are the drugs which have been reported to be resistant. Similarly, gram-positive cocci, common infections कौन-कौन से होते हैं? Staph aureus, strep pneumoniae, and इनके against इन drugs की resistance has been reported. Gram-negative cocci में Neisseria, penicillin G की resistance and fluoroquinolones is increasingly being reported. Okay, so that's another important table. Now the genetic basis of resistance, मैंने आपको बताया चारों mechanism. So at the start I told you 4 important mechanisms which are involved in generation of drug resistance. All of them include genetic changes. Okay, so let's have a word about the chromosomal mediated resistance. Chromosomal resistance is due to mutation in the gene that code for either the target of the drug or the transport system. अब ये concept आपको समझ में आना चाहिए मेरे earlier discussion से कि either the bacterial target is changed or the receptor is changed. Okay. The treatment of certain infection with 2 or more drugs is based on the following principle. If the frequency that a bacteria mutate to become resistant to antibiotic is 1 in 10 million, and the frequency of the same bacteria mutates to more resistant antibiotic is 1 in 100 million, then the chance that the bacterium will become resistant to both antibiotics is the product of the two, which is then a very, very rare phenomena. So this calculation is not important for you to remember. The idea is if the bacteria is resistant to drug A and then it is resistant to drug B, co-administration of the drugs will reduce the chance of resistance. So that's how you use both the antibiotics, and therefore it is very highly unlikely that the bacteria will become resistant to both the antibiotics. Okay, so multiple drug regime का ये एक important principle है. And you know कि if we are treating tuberculosis or if we are treating some serious infections which are of chronic nature, we usually give multiple drugs. It's never a single therapeutic regime जिसमें एक ही drug हो. We try to use different drugs, isoniazid, rifampin, you know all these things. Okay. So, what is plasmid-mediated resistance? Plasmid-mediated resistance is very important from a clinical point of view for 3 reasons, which are as follows. Number 1, it occurs in many different species, especially gram-negative rods. It frequently mediates resistance also to multiple drugs, so that's a problem. And plasmid have a high rate of transfer from one cell to other. So therefore, एक bacteria में अगर एक plasmid insert हो चुका है, it will be transferred to another bacteria. So many of the bacteria of the population will be resistant. Resistance plasmid R factors are extrachromosomal circular double-stranded DNA molecules. That's what they are. They carry genes for a variety of enzymes that degrade antibiotics. Okay, you don't have to worry about particular gene. Just remember there are drug-resistant genes. They are more so in the plasmids, which are extrachromosomal DNA. Resistant plasmids, most resistant plasmids have 2 sets of genes. Number 1, resistant transfer genes that encode the sex pilus and the other proteins that mediate transfer of the plasmid DNA during conjugation. So this is how the resistance in one bacteria is transferred to the other. Drug resistance genes that encode proteins that immediately either destroy the drug or change the targets. Okay, so simple stuff है ये. अगर आपने मेरा initial 2-4 मिनट का discussion गौर से सुन लिया, then you are good with the rest of the chapter. Now, R factors can carry 1 or more antibiotic resistance genes. Usually, plasmids से associated जो resistance है, that is for multiple drugs. That's the whole bottom line of this particular paragraph. Then there is Transposon-mediated resistance. These are the genes which are transferred within or between the larger pieces of DNA, such as bacterial chromosomes. And these are basically known as the jumping DNA. So if this is the bacteria and this is the chromosomal DNA and there is a plasmid, so the jumping transferable type of genetic material is known as the transposon. A typical drug resistance transposon is composed of 3 genes flanked on both sides by shorter DNA sequences. Not important for you to remember. ये याद रखने की ज़रूरत नहीं है. The 3 genes code for transposase, which is an enzyme that catalyzes excision and integration. Because these are jumping, अगर plasmid से निकल रहा है और chromosomal DNA में जाके attach हो रहा है, so it needs an enzyme जो उसको यहाँ से cut करे और यहाँ integrate करे. So इनमें इसकी genes होती है. Number 2, a repressor that regulates synthesis of transposase and the drug resistance gene. So इसमें भी कोई इतनी rocket science नहीं है, ठीक है. Now specific mechanisms of resistance, although ये हमने अभी ऊपर again मैं आपको बार बार कह रहा हूँ कि if you have listened to the initial 4-5 minutes of this video, you kind of grasp the concept, okay? अब उसकी details आ रही हैं सारी. So penicillins and cephalosporins में what happens कि if this is a bacteria and penicillins and cephalosporins enter, the usual mechanism is that the bacteria produces beta-lactamases and the beta-lactam ring is destroyed. अब ये जो बीटा लैक्टिमेजेस हैं, they have different properties. For example, staphylococcal penicillinase is inhibited by penicillin and is secreted outside the bacterium. In contrast, some beta lactamase produced by several gram-negative rods are constitutively produced, not important for your examinations. The beta lactamases produced by various gram-negative rods have different specificities. Some are more active. Okay, I mean, this is again not adding anything. ESBLs inactivate extended spectrum cephalosporins. All right, so the whole paragraph is telling you, all these 2-3 paragraphs, that specific mechanism में पहला mechanism इन्होंने बताया beta lactamase. Again, this is the mechanism number 1 जो हमने सबसे first table में discuss किया था. So you have to pay attention to this particular table. The first mechanism is production of enzyme which cleaves or inactivates the drug. Okay, penicillins being their example. So this is all too much, and then there are penicillin binding protein, then there is poor permeability. ये mechanism भी हम कर चुके हैं. So I mean, go through them and go through these paragraphs. हर drug की इन्होंने कोई ना कोई mechanism बताया. So if we are talking about penicillins and cephalosporins, usually beta lactamase is the one, and another one is throwing them out of the cell. Then and poor permeability, not allowing them to enter into the cell. If we talk about carbapenems, it is, और इसकी example क्या है? Imipenem. It is usually the carbapenemase bacteria produce करता है, which degrade the beta lactam ring again. Okay. If we talk about vancomycin, it is usually caused by change in the peptide component of the peptidoglycan from D-alanine to lactate. ये अभी हमने ऊपर example देखी थी कि the way the target is changed, तो vancomycin can no more act. Aminoglycides, there are multiple mechanisms of resistance. Modification of the drug by plasmid-mediated phosphorylating system and chromosomal mutations and decreased permeability. Tetracyclines की अगर बात करें, so basically ये सारा जो drugs के नाम इन्होंने दिए ना, it's all associated with this table. So if you, and we actually did this, if you have gone through this table, this is described in text. So what they have done is picked up this drug, for example, rifampin, and then they are telling you that rifampin में जो resistance मिलती है, how it works is that there is a change in the RNA polymerase and the target of the bacteria is actually changed. So rifampin actually cannot work. So let's go and find out rifampin and try to figure it out how this is working. So rifampin, for example, ये drug अगर आप bacteria को दे रहे हैं, so bacteria is producing a different target now. And if the different target is being produced, वो कैसे, वो target क्या है? वो target है RNA polymerase, because this is against which rifampin work. And if the gene of the RNA polymerase is changed, now the bacteria is producing a different type of RNA polymerase against which rifampin will not work, and hence the development of rifampin resistance. Okay, so read this text with the table that we discussed earlier. There are also some non-genetic bases of resistance. There are several non-genetic bases. For example, bacterial abscess can be challenging for cells and molecules to penetrate effectively. And this is very easily understood कि if there is an area जहाँ पे abscess है, तो obviously drugs cannot enter there. And it then becomes difficult for a drug to work on the cells which are present in the middle of an abscess. Okay, so simply the drug cannot reach there, and therefore they will be resistant to the drug. Bacteria can be in a resting stage. So if there's no growth in the bacteria, drugs are not going to work. Okay, the presence of foreign bodies such as surgical implants and catheters or other penetrating material makes successful antibiotic treatment even more difficult. So if the patient fails to take the drug, this is a compliance issue. अगर आपने patient को drug prescribe कर दी और घर जाके वो डब्बे में फेंक देता है, लेता नहीं है drugs, खाता नहीं है, so again there will be resistance. The drug is not working. So these are also some issues which you have to consider when you are considering a patient of resistance. आपको ये देखना पड़ेगा कि compliant है patient या नहीं, patient सही से drugs ले रहा है या नहीं ले रहा, कोई एब्सेस तो नहीं है, कोई फॉरेन बॉडी तो नहीं है. ये सारे कंडीशंस आपने कंसिडर करने हैं. सो बिकॉज सिर्फ आपने ये अज्यूम नहीं करना कि ये सारे मैकेनिज्म जेनेटिक मैकेनिज्म स्टार्ट हो गए बैक्टीरिया के अंदर एंड चेंज द ड्रग. मे बी द पेशेंट इज जस्ट नॉट कंप्लायंट, यू वांट टू चेंज द कंप्लायंस बिहेवियर. एंड इसीलिए बाज इन्फेक्शंस के लिए फॉर एग्जांपल डॉट से स्टार्ट हुआ है, डायरेक्टली ऑब्जर्व थेरेपी, कि एक बंदा जब दवाई ले रहा है तो उसको दूसरा बंदा विटनेस कर रहा है कि वो वाकई में दवाई ले रहा है या नहीं ले रहा. इससे मेक श्योर करते हैं कि कंप्लायंस एड्रेस हो रही है. Okay. Now, selection of resistant bacteria by overuse and misuse of antibiotics. There are several reasons why overuse or misuse of antibiotic enhance the selection of resistant mutants. The prescribing multiple antibiotics when one would be sufficient, prescribing unnecessarily long course of therapy, using antibiotics in self-limited infections, they all lead to increased chances of resistance. And this is why we strongly condemn that patients start, self-prescription and they prescribe themselves antibiotics. कि लास्ट टाइम जनाब मुझे इन्फेक्शन हुआ था, मैं डॉक्टर के पास गया था, इसी तरह मुझे खांसी थी, उसने ये एंटीबायोटिक लिख के दी थी, अभी मुझे वही एंटीबायोटिक खुद ही से ले लेता हूँ. भाई ऐसे नहीं करो प्लीज़. A physician will prescribe you based on so many different things. तो प्लीज़ गो टू द फिजिशियन यार, खुद से ना बन जाए डॉक्टर अनलेस आप खुद ही डॉक्टर हो. ओके, बिकॉज overuse and misuse of antibiotics lead to resistance. In many countries, antibiotics are sold over the counter. I mean, बहुत सारी कंट्रीज़ में इट इज़ ओनली बाय प्रिस्क्रिप्शन. बट इंडो पाक रीजन, ये जो हमारा साउथ ईस्ट एशिया रीजन है, यहाँ पे एंटीबायोटिक्स मिल जाती हैं फार्मेसी से विदाउट एनी सर्टिफाइड प्रिस्क्रिप्शन. यू कैन गो एंड टेक एनी एंटीबायोटिक यू वांट. सो इस वजह से मिसयूज भी ज़्यादा है एंड इस वजह से हमारे यहाँ रेजिस्टेंट स्ट्रेन्स भी ज़्यादा हैं. एंटीबायोटिक्स आर यूज्ड इन एनिमल फीड टू प्रिवेंट इन्फेक्शन एंड प्रमोट ग्रोथ. दे सेलेक्ट फॉर रेजिस्टेंट ऑर्गेनिज्म इन एनिमल्स एंड may contribute to the pool of resistant organisms in humans. So if we have a pool of resistant organism in the community, it's a problem. Okay. Antibiotic sensitivity testing, very important concept. Antibiogram is the term used to describe the result of antibiotic susceptibility test performed on bacteria which are isolated from patient. ये हर laboratory, हर hospital अपना antibiogram मेंटेन रखता है. इसमें बेसिकली ये बताते हैं कि वेल दिस इज द नंबर ऑफ केसेस दैट वी गॉट, दीज आर द बैक्टीरिया दैट वी गॉट, एंड एक्स नंबर ऑफ बैक्टीरिया ऑफ दिस पर्टिकुलर कैटेगरी वर सेंसिटिव अगेंस्ट दिस पर्टिकुलर ड्रग, देन दे वर नॉट सेंसिटिव अगेंस्ट दिस पर्टिकुलर ड्रग. सो यू हैव काइंड ऑफ अ सेंसिटिविटी पैटर्न प्रोफाइल. बाज़ हॉस्पिटल्स ये मंथली एंटीबायोग्राम मेंटेन करते हैं, बाज़ 6 मंथली रिव्यू करते हैं, बाज़ ईयरली रिव्यू करते हैं, टू देन हैव एन आइडिया कि विच टाइप ऑफ ऑर्गेनिजम्स आर प्रेवलेंट इन देयर पर्टिकुलर सेटअप. Okay, we don't have to go into the technical details how laboratory works up for that. Okay, this is all not asked again. I'm not going into the MIC concentration and minimal bacterial concentration because these are purely laboratory based stuff, but this is how we actually do the sensitivity testing for various antibiotics and for various organisms. Okay, right. I think that's all about what I had to talk about the resistant patterns. These are the summary boxes repeating the same thing. Just one thing which I would like to one more time highlight is use of antibiotic combinations. In most cases, the single best antimicrobial agent should be selected for use because this minimizes the side effects. Obviously, if you're using like 4 antibiotics, they will have more side effects. However, there are several instances in which multiple drugs are used. So for example, to treat serious infection, or particularly when the organism is known, you know that this is the drug profile that we have got to work against this particular organism, and to achieve synergistic inhibitory effect. So if you know that this organism will behave more nicely if you use these 2 drugs because they work synergistically, they will enhance each other's effect, then you can use multiple drugs. And to prevent the emergence of resistant organisms such as in tuberculosis, you don't want a resistant organism to develop in tuberculosis, so use multiple. So these are few instances, और ये एग्जाम में बहुत पूछते हैं कि when would you combine antibiotics. These are the 3 usual scenarios where you do this. Okay, now ये मैंने बिल्कुल ऑफसेट छोड़ दिया था, बट आई थिंक इसकी डेफिनेशन एटलीस्ट मैं आपको बता दूं so that we complete this chapter. Minimal bacterial concentration for certain infections such as endocarditis, it is important to know the concentration of the drug that actually kills the organism rather than the concentration that merely inhibit the growth. So here the purpose is to kill the organism because of the severity of the disease, and this concentration is called the minimal bacterial concentration. Is determined by taking a sample in the tubes, and how we calculate is not the issue. What minimal bacterial concentration की definition क्या है, that is an important issue. So what you need to know is that what is the concentration of the drug which will actually kill the organisms present in that particular patient. That is called minimal bacterial concentration. And MIC, for many infections, the results of sensitivity testing are important in the choice of antibiotic. These results are commonly reported in a unit, a reporting unit, MIC, which is defined as the lowest concentration of the drug that will inhibit the growth of the organism. So here we are talking about inhibiting the growth of the organism. Here we are talking about the concentration of the drug which will actually kill the organism. So these are the two basic units that we use in laboratory to actually report कि drug देनी कितनी है. MIC is about inhibiting the growth curve of the bacteria, and minimum MBC is about what is the concentration of the drug required to kill the bacteria. Okay, so with this, these concepts are more sort of discussed in pharmacology. What is synergism, antagonism, and indifferent behavior? Synergism is basically when you are giving multiple drugs and they are augmenting each other's effect. If they are cancelling each other's effect, then this is antagonism, and if they're not doing anything to each other, that is indifferent behavior. So that's all about this chapter. आपसे मुलाकात होगी अगली वीडियो में बहुत जल्द. अपना ख्याल रखिएगा. My name is Professor Asif Qureshi. You are watching Dr. Asif Lectures.